All right, good morning, everyone. It is my pleasure to introduce the speaker today to you. His name is Ofer Levi. He's from the University of Toronto, and he's going to talk today about integrated sensors and nanostructures for biosensing and biomedical applications. I've been knowing Ofer for quite a number of years. He joined the group at Stanford when I was a postdoc myself in the same group. Ofer was coming at that time from Jerusalem. He was getting a bachelor's degree from the Jerusalem College of Technology and then a PhD from the Hebrew University. And then he went to Stanford and had various positions there. And then since 2007, I believe, he's a professor in the electrical engineering department at the Institute for Biomedical Engineering at the University of Toronto, and I'm looking forward to his presentation today, so please hold me.
Good, thank you very much. So what I'm going to talk today is about could you hear me in the back? Good. So I'm going to talk about today is about integrated sensors and nanostructures for biosensing and biomedical imaging. So in this regard, what we're going to talk is both about micro and nano devices using semiconductor technologies and see some of the directions we think that are interesting in trying to get these devices for biomedical care. So let's get started. Micro and nano fabrication, I'll give you some examples for what has it already revolutionized. It has revolutionized computation. Also imaging. DALSA is a nearby company, and their achievements in imaging are well known. So this is another example. Telecommunication, Canadian industry in telecommunication is also well known. And all of these achievements were enabled just because of the micro and nano technologies. And much more if you think about solar cells and all the investments that are happening now around the globe to try to have micro and nano technologies to improve efficiency in the solar cells. So all of these technologies are available, and part of our efforts are trying to think about them and how do we use them for biomedical applications. So here are some of the examples, as you could see, with biology and medicine, where you have DNA microarrays. You could think about drug delivery, or maybe patterning neural cells that they would conform to a very specific chip. So all of these examples are examples for using micro and nanotechnologies. I'll touch a little bit on the issue of microfluidics and why do we see in many times microfluidics as synergetic to micro and nanotechnologies that we use for sensing. But all of this has already been done, and now we would like to take it a step further and use it also for devices for healthcare. So what we're hoping in general is to describe a process with biosensing. So what my lab is doing is focusing on developing technologies and devices for biosensing. And you could see over here on the top a schematic of a step-by-step process that you would find when you have biosensing. So you would start with sampling, where you introduce a sample for the sensing element if you have a small device. Then you have a biorecognition. There usually is some element, whether if it's an antibody or an enzyme, that sits on that surface, and with that element, it recognizes the analyte, and just that analyte and nothing else. So this is the second step. Then there is some stimulation, where externally you send light or an electrical stimulus, and with that you're trying to extract the information from the analyte. So this is the first step. Then we have signal transduction, where the information that was there that we tried to read with either optical or electrical signal is being read back to the sensor. And the last part is the detection, where you acquire the desired signal. Now, obviously, this is a very schematic thing that I used to introduce students when I teach my classes, but in reality, many of these components are merged, especially when you talk about micro and nano scale. Because are you really delivering a sample, or in the same time delivering sensing and then moving it to the right place? So all of these components, especially when you think about micro and nanotechnologies, are merged together. But I'll try to give some examples as part of the introduction before I'll go into what we do. So the sampling part, this is a microfluidics from Steve Quake's group, where you could see many colored images of dyes that are food dyes, and you could see the intricate circuits in the middle. So in their lab, they're trying to develop technologies with very advanced microfluidics and pumps and such that would allow you to have many experiments done in parallel. So parallelism is clearly an advantage with thinking about sampling, because if you want to work in the micro and the nano world, you would like to do the same experiment many times over in parallel. The next step that we talked about was biorecognition, and this is just an image of a classic description of an antibody, where this is used as the capture mechanism. When you have an unknown sample coming in and you're only looking for one analyte, whether it's a virus or a specific protein you care about, having a capture molecule that only has affinity or ability to recognize that very same element is a key issue. So this is many times an active research in chemical engineering and working together with chemists to try to find the right capture molecule or antibody. The next step, which tries to mix into detection, is the biorecognition. In our case, we try to use it optically, so you would have some kind of a laser detector combination that would send the light into the microfluidic channel, and then the analytes that are being captured on the antibody in this schematic over here, they will be red because there will be some signal change, whether it will be fluorescence signal or maybe index refraction signal. And I'll try to give example of both cases because we have worked on both application. So this is an example for the biorecognition where you have a readout, and a lot of efforts are made in the micro and nano world to make this component, both the microfluidics and the detection, to be very compact. So this is part of the many efforts that are done in the field. Now, in order to stimulate, this is some examples with readout where you have a finger prick and you try to have glucose readout in one example, or perhaps readout from the body. This is an example of a mouse, and as you could see here, what you see here in green is mostly autofluorescence, the natural fluorescence from the body, but they want to read out these two fluorescent dyes that usually are targeting either stem cells or perhaps targeting a tumor. So the idea of stimulation is you send the light and you would like to get information from these very specific elements. So depending on your technique, you either do it electrically or optically. We like to use optical technologies because it is non-invasive. You send the light, you do not disturb the biology, and you can get the readout back. So this is part of why we believe it's really advantageous. So this is one thing, and then taking it further, and I'm going to only talk a little bit about this aspect, but if you want, you could talk to me afterwards. One of the challenges we thought was, in addition to healthcare and diagnostics, many people are doing studies where they take molecular imaging, and this is an example for an MRI on the right, and on the left, optical imaging technologies on mice, and the ability to take snapshots of the dynamics, especially molecular dynamic, already exists. So this is one way of taking information in snapshots. On the other hand, there are these very limited technologies that allow us continuously to take information about the body. If you want to think about cardiac monitoring, there are systems which allows you to have EKG and other systems constantly on the body. So we're thinking about the question, what about continuous molecular imaging and monitoring in live animals? Can we take our technologies that are able to be miniature because we use micro and nanotechnologies and make them portable enough so you could think about reporting constantly and continuously? And when you think about reporting constantly and continuously, it becomes a tool that is beyond just diagnostics. You could think about monitoring therapy. You could think about analysis that looks into the time dynamics as a key issue, and not so much do I get more light here than the light there. So in our lab, we seek to develop miniature sensors and sensing technologies that are based on light that would allow us also continuous imaging and monitoring of live animals. So in addition to what I'm going to focus today on lab-on-a-chip and diagnostics, our lab are taking the same technologies that we're working on with micro and nanotechnologies and trying to think how do we use them in a very smart way within live animals. So the building block that we have worked in the last few years with collaboration of several people, but mainly with Professor Jim Harris at Stanford, is this thing you could see on the left. This is the tiny sensor. This is how big it is, and it's really tough to go to the clean room and then find it at the end once you diced it. But this is a very small element. This is a zoom in of the sensor. It has about 10 or so layers that comprise the lasers, the detectors, and some intricate contacts and layers around it that are supposed to isolate one from another. So this is the building block that we're using, and as a building block, it allows us to do readout of light. So we send light from these lasers in the middle, and then we read it in the same element with the detector. So I'll tell you a little bit about these building blocks, and I'll give you an example how we use it for sensitive sensing, and then I will go and talk about the biosensing within microfluidics in another example. So the first example I'm going to talk about would be about how do we use these very small and miniature building blocks. As you can see, they're really tiny. And how do we use them in order to do fluorescent measurements? So you send light in. There is some fluorescent tag or fluorescent marker in the tissue or in the microfluidics we care about, and then the reporting is of fluorescence. So usually we have some challenges in how to do it, and I'll describe a little bit about it. The building blocks we're working on are based on vertical cavity lasers. So these are vertical cavity surface emitting lasers. The schematics is shown here on the right, where we have the bottom DBR as the distributed Bragg reflector as one mirror. We have the top part, which is another mirror, and there is a gain region in the middle. So as you could see, the architecture is such that the laser emits upwards. So this is why it's called vertical cavity surface emitting lasers, because the architecture is such that the light is going upwards. And there are many, many advantages both in manufacturing as well as in making the devices and using them that makes this very, very attractive. So we've developed the devices, and they have about 1 milliwatt or more at 670 nanometer. And this is a very interesting wavelength because many of the biologists are used to work within the visible light. So this is just the top edge of what they're used to in the red where they do many of their studies, whereas this is more or less the bottom edge of where semiconductor technologies have ability to create light sources. So we try to merge them around the wavelength of 670 nanometer. Why do we like vertical cavity surface emitting lasers? So first of all, almost any mouse that you have, including this mouse, has a vertical cavity laser inside it. This is the main laser that is used and is made in millions and millions every day for this process. So we like these technologies because it is scalable and robust. You could make a wafer with 10,000 lasers easily every day when you make these devices, and sometimes we work with startup companies that work with vertical cavity lasers. So sometimes one wafer that they make is enough for 2 months because they have so many lasers on them that they could make many, many measurements and then 2 months later go again. It allows us to have multiplexing and multimodality because we could make measurements not only on fluorescence, we could measure absorption, we could measure Raman sensing. It is a very flexible way if you send a laser light in, which is coherent. On top of it, and this is part of the studies that Elizabeth Monroe in my lab did in the last year, was how do we manipulate the vertical cavity lasers to make them much less coherent, to behave like a light emitting dad. And there are huge advantages to manipulating the coherence. It is very low cost and disposable. I think in mass production, like here in the mouse, it costs less than $5 a piece for the whole unit, and it has a very high performance. The systems that employ vertical cavity lasers with speeds as much as 10 gigabits are being produced. So it's a very scalable and robust technology that we like to use. Now, if you want to apply them for sensing, there are some challenges. And I'm going to focus on sensing, like in this diagram I showed before, where we have a flow of analytes that we would like to capture, and they have some fluorescence recognition, and we're trying to use them to make a fluorescence measurement. So what are the building blocks that we want to think about? The first one that we think about is the laser. That's what we just talked about. So the vertical cavity laser is shown over here. These are these two little rounds in the center. For redundancy, we always try to make two and not one, because many times we would burn a laser in the middle of the experiment, and the last thing you want to do is just stop and go home. So we always like to have at least two lasers in the same time. These lasers are at 670 nanometer because this is the wavelength that is optimized for using with the dye we chose. The dye we chose was Psi-55 that has advantages in very low autofluorescence in the body as a background. So this is the laser that we chose, and it's shown here in the middle. But the majority of the areas you could see is taken by the detector. This is good because every time we want to make a smaller detector, we could make much more dense array. The reason we have about 1 millimeter square or so of detector is to allow us a very high sensitivity. And we got about 1 to 2 picoamp, depending on the graduate student in the processing in the clean room, in the processing capability. But that took a little bit of time to perfect to get very low dark current. So we have a very sensitive detector with a powerful laser. The only thing that is left is what's shown here, and you cannot really see it, but there is a 7-micron layer of optical filter just covering all the detector. And the purpose of that filter is to block the laser light while allowing fluorescence in. Any fluorescence detector blocks the laser light and allows fluorescence in. And normally, fluorescence is about 5 orders of magnitude lower in intensity as compared with the laser. So it's a real challenge to allow us to block. So the design was about 10 to the 6th in blocking. And with all imperfections, it's not as good, but you have to work very hard to design a filter that absolutely blocks all the laser. And there are lots of things that I would not go into details, but if you want to ask me how do you get to these details and how do you really make it, well, we've just submitted a paper to Optics Express that describes how do we get and achieve these. And there was an earlier work by a graduate student that worked with me, Evan Thrush, that had an earlier generation at 780 nanometer. The topology is very similar, but the excitation wavelength was at 780 nanometer. So we have the capability to choose several wavelengths when we try to use these building blocks. What are the components of the sensor? We talked about the laser, so let's see what happens afterwards. We start with the laser. We shine light into the biology, and it could be either a tissue in the case of a live mouse, or in the case of a flow channel, there will be fluorescent elements that flow around in the microfluidics channel. So we shine the laser light in, that's shown in blue, and that creates fluorescence. Fluorescence goes into everywhere because it's isotropic. So some of it is going backwards towards the detector, as shown over here. So we collect the light above that reaches here using a PIN diode. So the PIN diode is what is here shown as part of the detector. In the same time, we need to block the backscattered light, which is why we have that emission filter we talked about. So the emission filter has the objective to remove the light from the laser while allowing the emission of the fluorescence to come in. So what we're going to see is a process where we very efficiently excite the light to the biology and then collect fluorescence from it. And hopefully the emission, if you design it right, the emission will go through. So the excitation is blocked by the filter while the emission is going through. So this is a miniaturization of a very classical fluorescence microscope that you would find in many, many labs. Take the whole microscope and shrink it to a 1 millimeter cube. But we keep all the building blocks as we do that. So we chose to work with this dye, as you could see here in the diagram. The excitation that we chose is around the peak, which is around 675 nanometers. So this is where we optimize the laser light to be. And we create an emission that goes well above 700 nanometers. So in this case, for the biology we're looking to collect light and emission above 700 nanometer. And the reason we like infrared imaging is because that we could penetrate very deep into the tissue or any biology we care about, because you could see these are the optical properties in these wavelengths. A very low absorption. Most of what you lose is due to scattering. So if it's scattering, you could penetrate a millimeter into tissue, and this is very easy to collect afterwards. In the same time, the autofluorescence, the natural ability of the body to create fluorescence, which is the background, you would like not to see the background, is rather low. So in comparison to the visible, where you could see glowing mice that are glowing in the green, this doesn't happen. So these are part of the advantages of why we chose Sci-Fi 5 dyes. I'll jump forward after about 2 years of fabrication and optimization and many, many hours in the clean room. These are the results of the sensor. The good news is that the sensor, I think, now is about within the category of integrated sensors within the best in the world for fluorescence. In sensors that combine both lasers, detectors, and the microfluidics. So what you see over here is the detection, and this is a photocurrent in picoamp, and this is the sensitivity in nanomolar. Note that in about 1 picoamp of current, we put a background variation limit. It is not that the sensor is not sensitive to pick up below a picoamp, but every time you change, every time you move a cable, if the system is not stabilized, any slight movement is about 1 picoamp or so of current changes. And this is why we have some ambiguity that we're not sure if what we see is real or maybe the background changing. So we have a background variation that limits us to about 5 to 10 nanomolar sensitivity. And this is quite good if you think about the sensitivity in the context of a system that has everything within a millimeter cube. So this is quite an achievement. We were very happy with it. And it is really limited by the background, not by the sensor sensitivity. Then we try to do the same thing, but on a mouse. And this experiment, this is done in collaboration with Sam Gambir in his lab. You could see these are 2 lasers, and you could see here it's shown in blue, but usually in my laptop it's shown much more as red, which is the laser light. So you shine the laser light in. This is the package for the sensor. In order to package it for robustness, we keep it in a standard TO can. And then you could see, so you have one place where you want to measure and the other place where you have a control. Because if you have some movements, you would like to do it. And then you could measure it and plot the concentration on this axis while measuring the photocurrent. So in this case, we have a higher background. And this background is about 10 picoamp and is due to the tissue. So again, this is not limited by the sensor by any means, but the fact you have a live tissue and you have backscattering of the light. So this is what limits us to this sensitivity. And we get a really nice straight line between what we expect and what we get in the measurement. If we compare it to a giant system called IVIS made by Caliper and Xenogen, it's about $200,000 to $300,000, and we have 2 of them now at the hospital at Princess Margaret that we work with. So you could correlate what you would get from this CCD-based system on the y-axis and our sensor on the x-axis, and you correlate the concentration. And as we could see, we have a very good agreement. So in both cases, both the very expensive system and our tiny sensor, the limitation in making the measurement in fluorescence sensing is not by the sensor. It is by the tissue backscattering that really puts a higher bound. So we thought that we said this is good because it is always good to be in a point where you're not limited by the technology, you're really limited by the biology. So we went ahead and we did some comparative studies, and this is what you see in a picture in that CCD-based system I was telling you about, IVIS. So what you see here is a glowing mouse due to autofluorescence. And then on the back side, we injected a small amount of dye. And that allows us to know what is the sensitivity. So you could see that if you inject a dye with a concentration of 50 nanomolar, you could see it. But if you inject in about 25 nanomolar, you could barely see it. So the biology and the autofluorescence at that wavelength that we work with, where the biologists like to work, is limiting the sensitivity inside the body to about 25 nanomolar or so, which is again a biology-driven issue. It's very much matching literature results. This is a result from a group in Texas. As you could see, they got very similar results in seeing the whole body of a mouse glowing, and this is about 50 nanomolar of concentration. So we get very similar results to what other groups have measured as the limitation of autofluorescence. And the good news is that the sensitivity is not limited by technology. So if we choose to find an application where we need more sensitivity, in that case, we would probably move to longer wavelengths, and we would need to develop a little bit more biology. But in this wavelength where the biology is well developed, we could get only that sensitivity. Here are some examples of the measurement system. It's based on some lock-in amplification as a step of making all the measurements off chip. We're now working on making it simpler, but this is the system as it is. And this is a zoom in of the two sensors and the BNCs going out. The main effort here was really to make it robust, because every kink on the cable would send you a picoamp of fluctuation, and in this sensitivity it really matters. So we did some studies to see, as preliminary sensing, to see what could we see when we inject a dye into a tumor. And as you could see, we have here on the left time. So this is a few hours of recording, and this is the intensity. And as you could see, we're measuring in parallel the left and the right side of the mouse. So one is the control side that never saw a tumor, and the right side saw a tumor. And as you could see, there is a difference between the tumor and the control. Now, if you plot the ratio between them, it's about between 2 and 3. And this is a limitation of the type of dye and molecular marker that the biologists are using. The ability to differentiate a tumor from a non-tumor is limited by the chemistry, and there is a lot of effort to get it better, but even with that, we could still distinguish very nicely between them, even with that limited biology. And this is an example for continuous measurement. So if one would like to try, for example, to evaluate a drug, you could watch it very rapidly to see, is this drug working or not as you're targeting a tumor. So this is an example where the temporal events do make a difference. So this is an example I wanted to give. That is one example. We have developed a sensor for fluorescence. We have used epitaxial layer growth in nano and micro lithography, and then later on we were able to make functional laser and detector all into a very small sensor unit, and this is an example of application. From this point on, I'll move to the second part of my talk, when we'll focus more on nanosensing, because this is an example where we used fluorescence sensing. In the second part, I'll try to focus more on index of refraction sensing that we use in the nanoscale. And depending on the time, we might go into details or go a little bit as an overview. So part of the motivation to work within microfluidics and diagnostics is this example of what happens in the developing world. We're in Canada, and we're fortunate to have very advanced hospital system developed. Yesterday, there was a big debate in the Congress in the US about introducing a similar system to what we have here. But in many, many places in the developing world, this is how a hospital lab looks like. And this is not very encouraging. So if you have an opportunity to have very small miniaturized system that do both sampling and detection, that would greatly help many, many people, because it could lower the cost in making measurements. So there is a long history in the last 20 years of working on miniature technologies for portable diagnostics, and this is part of this effort. If you think about biosensors and microfluidics, so this is an example of a 4-inch wafer with microfluidics, where you have many, many sampling areas, and then in the region is where you have detection. Why are these appealing? So these are some of the examples for the applications, but there are many, many advantages that make it very attractive. It is allowing you real-time measurements. It has a high sensitivity if you design the assay right, because it has a very small amount of volume. It has a low detection limit, because you could isolate the measurement from many, many other elements. The sample volume is small. Nobody would take a milliliter of blood or 5 milliliters of blood from you every time you need to make a sample. A tiny finger prick would be good enough. if you work on technologies that do not have fluorescence, it could also be label-free, and I'll talk about the aspect of label-free in a second. It has a high throughput. As you could see, you could run many, many samples in parallel in such a microfluidics design, and it is very good for point-of-care testing, both in developing worlds as well as in North America. So we like these technologies a lot. If you think about integrated optofluidics, it is a field that tries to take both the optical detection and merge it within the fluidics element. So that way you could integrate both microscale and nanoscale technologies in order to make these applications. So this is where we're trying to work in this field within the optofluidics. So this is an example from a review by Ben Eggleton's group in Nature Photonics from 2 years ago, where you have some, in this example, it's a photonic crystal element, and this is the microfluidics, and you could measure the difference between one analyte and the other analyte as it goes above the detection system. So above is the microfluidics layer, and below is the optical layer. And he gives some examples of sensing technologies that each one of them uses micro and nanotechnologies. You could think about waveguide designs, which are optical waveguides. So you see a host track design for a waveguide, where every time you change the index, the coupling between the waveguide and this host track will change. You could think about a toroid type of design where we create whispering gallery modes that are optical modes around this region. So every time an analyte comes in, it changes the whispering gallery mode conditions. And also these are other examples, and this example is using planar photonic crystals. So we will talk a little bit about photonic crystals as a way to use detection. So this is within the field of optofluidics. As we mentioned before, we have focused on techniques that are mainly fluorescence. But fluorescence is not the only mechanism we could use. You could think about absorption and index of refraction when you shine light, and the attenuation or change of index could be measured. You could also measure scattering, where either you measure intrinsic properties or maybe exogenous elements like a particle, a gold nanoparticle you would put. So within optical measurements, whenever you think about biodetection, and this is a slide I got from Chris Contag at Stanford, you always could go down to say, what is the mechanism I'm trying to optimize? So in this case, the advantage of thinking about maybe absorption and index of refraction sensing is that these are label-free techniques. The analyte by itself would create this change in absorption and index of refraction, and it would allow us to have a label-free detection. Once it's a label-free detection, you save time, you do not disturb the biology, things could be much faster. So within a diagnostic kit, if you could find a solution which is label-free, this is an advantage. So we gave an example of fluorescence sensing. In this case, we're going to talk about index of refraction sensing as the main example for microfluidics. We have worked already on fluorescence and got very nice results, as you saw before, but now we're going to talk about the other modality. So what is the principle behind index of refraction sensing? Normally, you create an optical resonance. If you measure the optical structure, you usually get some type of resonance. In this case, it's a Lorentzian resonance, but you could get many types of resonances. And whenever the index changes due to the environment, the resonance shifts. So what you could measure is the index of refraction change as the x-axis, and then you would see how much was that shift. And from that, you could deduce a line, and the slope of the line is the sensitivity. The principle behind it is that every time biology comes in, usually the proteins or the molecules would displace water. So you had a water molecule there, you take it away, and you would put a protein or a cell there, and this displacement creates an index of refraction change. Once you create an index of refraction change, it could be depicted here. Now, as you notice, the shift is not the only thing you want to think about. The other parameter is how sharp is the resonance. Obviously, if you have a very narrow resonance in the optical measurement, it is much easier to detect versus if it's a really large resonance and you have to spend many, many hours of measurement to say, I was able to measure this very slight element. So it's always a matter of how much could I shift it, and then how narrow could I create a peak. And these are the two measures we're going to talk about. And we're going to use the example of photonic crystal slabs as a way to measure index of refraction sensing. So the two metrics that I'm going to introduce, one is the bulk sensitivity. How much of a change in wavelength did I create by change of index? I remind you that the reason we get a change in the index of refraction was that an analyte came in and it displaced another molecule. Usually it's a water molecule. So that displacement created a very small index of refraction change. And you could see this by the shift. So the units are nanometers for a refractive index unit change, which is the units that you could quantify it. And this is one way to measure the sensitivity. The better the sensor, the higher this value would be. The other one is exactly the opposite. The detection limit is an index of refraction unit. So it is a ratio that says if I could measure 10 to the minus 5 change in the index, the lower it is, the better it is. And it is proportional to the delta omega, which is the full-width half max of the peak divided by the sensitivity. So the narrower the peak or the bigger the shift, the better I could get the detection. And there is some proportionality factor K that depends on the physics of the problem, because you could create a Lorentzian shape, you could create a Fano resonance, you could create many, many shapes of resonance peak. So the K factor has to do a lot with the shape of the resonance that you create. Altogether, that creates some merit functions that one could use in analyzing good, bad, did I get a good detection or bad detection. So these are the functions of merit we're going to look at. I'll give you an example which is very common in the last few years. This is an index of refraction sensors, and this is based on surface plasmon resonance. So what you see over here is that you start with a monochromatic light source, you shine it on a surface, and this surface usually has a dielectric meta interface, and in some angles you couple the light to surface plasmon polaritons. And in these angles you see a very specific notch in the intensity that is reflected. So if you have a photodiode array, you would see it. So what would happen if you see here on the left, if you would make a measurement, the beginning you would have this thing that is shown on the dark black line, where you had on top of the surface of the metal, this is the metal-water interface, the dielectric is on the bottom, and in the metal dielectric, say this is water, you would have an electric field that penetrates the water. So in this case, we have an electric field that penetrates the water, and because it penetrates the water, if you have antibodies, any change due to an analyte that is captured on the antibodies, as we see here on the bottom, any change would create a local change in the environment, and that local change of the environment is shown as an index of refraction change. So in that case, we would see a shift of the resonance peak. So what you measure is either an angular or a wavelength-based plot, as you could see over here, and whenever you see a shift, then you say, I've found the analyte. So this is a principle of surface plasmon-based sensors and how you use them to measure index of refraction. The typical values for that is quality factor of 50. So this is one of the biggest challenges with using these technologies, that the design here is based on metal surfaces. And metal surfaces, for the most part, are lossy. Because they're lossy, the quality factor that relates to how wide is the peak is quite low. So this is an advantage that has, if you could come up with a dielectric design, like, for example, with photonic crystals, where the quality factor could be much higher, or in other words, we could make a much narrower peak. The sensitivity, on the other hand, is quite high. Normally, if you want to see how good they are, just multiply the quality factor by the sensitivity. That would give you a good figure of merit. So now let's talk for about 2 or 3 minutes about photonic crystals and how do we want to use the photonic crystals as a sensing building block. So that field was started by Sajeev John from Toronto, as well as Ilya Ablonovich from UCLA, as suggesting we could create a periodicity that will be in optical materials that is very similar to what you find in semiconductors. And you could see that you could think about periodicity that will be in 1 dimension, 2 dimensions, or 3 dimensions. So depending on the periodicity, you could create very interesting effects in them. And if you think about it, you could create a building block that looks like a wood pile over here, which is very similar to a unit cell in a semiconductor. And just like last night, there was an announcement by a science magazine that John Pendry's group in London, as well as another group from Germany, found a way to use it for cloaking. So there was a metal layer, there was a tiny defect over there, and they were finding a way to use these technologies to hide that element. So these are examples of how you use them for many, many applications, including integrated circuits and trying to hide elements, as well as the more conventional ones like filters and pigments. What we're going to talk about is different. We're going to talk about photonic crystal slabs. As you could see, you could create a slab where you start with a slab, say in silicon or iron dielectric, and then you create a periodic structure of either holes or rods. Depending on the design, both of them are regarded as a photonic crystal. Now, this is quasi-2-dimensional because within any plane, the periodicity creates a 2-dimensional photonic crystal, but at the same time, the material itself creates some difference on the other dimension, namely the Z dimension. So over here, for example, if it's a silicon device, you have water above or water below, or maybe air above or air below, and that difference in the height creates effectively a 3-dimensional structure. So these create what we call guided resonances, the ability to couple light from the outside world to the inside of the element. So normally if you have an optical waveguide, you talk about guided modes. So you could create guided modes that would be inside them. But the biggest challenge for many people who work within optics and trying to launch light into a waveguide is, how do I get the light in? Guided resonances are unique cases in which I could couple from the outside world very easily to these guided modes inside the crystals. And that's part of the flexibility that photonic crystals allow. So if you think about it, what we're trying to do is to measure the transmission versus wavelength. And this is a typical result we get from a simulation. As you could see over here, if you measure the transmission versus frequency, you get these guided resonances that are these very sharp resonances in the spectra. The resonances come from the ability to couple light very efficiently into the waveguide. So these are examples of how very nicely I couple light in, and then they behave as guided resonances. Now, we could use these guided resonances for sensing, because what you could see is if you change the environment around the photonic crystal slab, in that case, these guided resonances would slightly move. So we have a high quality factor resonance, as you could see in these two examples. And not only did you have a high quality factor resonance, you also could use it for sensing. So this is what we're going to do. So we went ahead and fabricated some of these designs after we modeled them. And we have looked into two cases. In both cases, we have a periodicity of photonic crystals. So this one was using E-beam lithography. In the past, we also used optical lithography to create 2-dimensional arrays of holes. And the diameters of the hole could be anywhere between 200 nanometers to a micron, depending on your design. And the periodicity is between half a micron to a micron. So this is how it looks in an SEM, and this is how the whole structure looks in about 200 micron or 500 micron diameter. So that gives you a feeling for the structure that we make, and you could even see the moire effect, because even the SEM had some effects when we tried to measure it. And we tried to look for designs that either have a symmetric design where we have both water above and below the structure. So we create guided resonance within the structure. We like to use silicon nitride because it's transparent in the visible and transparent in the near infrared, and it's very flexible to work with. And we looked at a design that is all transparent with the silicon dioxide or quartz underneath as the substrate, versus a case where we totally removed it and saw that we could get a symmetric design. Now, we looked into the physics of it, and we saw a very interesting case for what we have for the guided resonance. And I wanted to spend a minute in describing them because we got two resonances, one which is usually in lower frequency, higher wavelength, that we describe as TE-like resonance, and the other is TM-like resonance, which is higher frequency, much narrower peaks. So this is an example, and it's not shown so well, but at least I'll do my best to describe. I'm sorry, the projector is not as nice as my screen. But what you could see over here is that this is the example of the electric field intensity. So you could see in the case of the TM-like mode, the light penetrates like a candle up and down to the water side. As you could see over here and on the bottom, most of the energy is outside. So this is really good for biosensing. If you create all the energy to go outside, it allows you to have a very good connection to the biology in the water. On the other hand, the T-like structure would have this one, which is mostly within the hole. So we get two types of designs. In one design, we have most of the light outside. This is the TM-like. In the other design, we get it very localized within the structure. So two options. We could have it localized within the structure, or we could have it penetrating. So it gives us a very large flexibility on the design and fabrication and using it. Also, and I'll give you some examples for how does it look like. So if you look about the electric field, these are the examples of electric field in X, Y, and Z. And for the most part, we launched light along the Y polarization. So we could see that there is a very good coupling of the light along Y, but very bad coupling in both X and Z. So in the T mode, we very efficiently couple light to this mode, and most of the energy is along the Y direction, not penetrating up and down. In comparison, in the TM-like, as you could expect from the energy plot, we should see a very dominant Z component. As you could see here, there is a very dominant Z component. The energy is not only along the Y, which is along the slab, but there is also energy that penetrates above and below. So we see it not only from the energy intensity, but also from the electric field. And the resonances are much sharper. We could change the resonance by changing the radius, so that gives us flexibility. By creating several radii when you fabricate a device, you could slightly move and tune where would you like the resonance to be. So this is very efficient. And another thing is that if you choose to have an asymmetric design, which is much more practical to do, where you keep a substrate and then on top of it you would have the silicon nitride. So what you could see is that you create asymmetry. Usually in any optical waveguide, the lower index gets less of the electric field. So as you could see here, already in the TE, you could see that most of the electric field sits in the lower substrate. But if you compare it to TM, you could see it much more dramatically. Because we have water above and silicon dioxide on the bottom, this asymmetry made most of the energy go down to the substrate, so much less of it is available to be used with the biology and analyte detection. So knowing the physics really allows you the manipulation to see how do we go all the way to electromagnetic simulations, either in analytical form or in a finite difference time domain, analyze the electric field and go back and say, how do we use these nanostructures for biological detection, where we want to localize the biological molecules either on the top or inside the holes. So I just wanted to give you some example of the simulations that are involved, because they really give us some insight. I wouldn't have too much time to go into details. I'll just briefly mention that if you compare, and this is a large table that tries to compare them, if you compare them, you could see that the TM-like materials that are shown over here gives us better sensitivity and better quality factor. So they're superior as compared with the TE-like material, and if you want, we could go maybe later into the details. But this is the conclusion of the analysis, that it's always good to work with a team like resonances when it comes to biosensing. Much harder to fabricate, because it is really hard to fabricate quality factors that are harder than 1,000 if you want to think about larger elements. But if you could do it, it's really worth the effort. Some examples of measurements we did in comparing, talking about designing something and then going to the lab and fabricating. Many times when you try to fabricate a device, it's not always the best. So this is some of our efforts that from 2 years ago, where you could see the simulated resonances. One is T-like, one is TM-like, and you can see that the resonances are not exactly matching. The location is matched, but the width is not as good. Usually this is due to disorders in the fabrication. So the better the fabrication, the more uniform is your device, the better you're going to get the sensitivity. It's always a combination of movement of the peak, which we call sensitivity, and also the quality factor, how narrow is the peak. So this is one example. But even with that, you could see we made a measurement where you could measure a peak moving by changing the index of refraction by blending water and IPA, and you could see this change. So that was a good effort that we did, and then we went back to design and make better devices. So lately, we made these devices with the new e-beam at Toronto, and this is the work that will be presented to the CLIO conference in about 2 months in California. So in these 3 examples, we compared different index of refraction of the water media. And what you see here, each one of the cases is the wavelength versus transmission. And one color is usually the simulation, and the other color is the experiment. So let's take this one as an example. You could see that the simulation predicted quite a very sharp peak in the T-like resonance, and in the experiment it became a split peak. So that's usually due to non-uniformity in the fabrication process. But the TM-like peak was much less influenced. And the reason is, if you remember, most of the energy in the TM-like peaks sits outside. It doesn't sit inside the hole. And therefore, if you have imperfections in fabrication that are shown mainly in the middle of the hole, TM-like peaks would much less be influenced by this. So this is an example where you could use that and still get a very good biosensing. And from the fit here, we could see that we get a very good match between the theory and the experiment in our devices. So these are microfluidics devices we make, and the theory and the experiment agree quite well. What are we going to do with it? And I would like to wrap up so we have some time for questions. We're going to evaluate the suspended symmetric devices to allow water above and below the structure. That will give us about 2 to 3 more times sensitivity. In the incorporation to PDMS devices, as I showed before, this is an example for PDMS-based microfluidics. So as we incorporate, what did I do? Sorry. So as we incorporate them, we would like to characterize the sensitivity of the device and compare it to the predictions, quantify what is the detection limit. We hope to get to detection limits that are beyond 10 to the minus 5 in a very simple and robust detection system. Demonstrate both the sensing and kinetics when we bring an analyte molecule in, and then watch it. We could watch the intensity change over time. And then we would like also to go back and try to optimize the devices, not for communication wavelengths, but back around 700 nanometers, where we could do a measurement both of fluorescence and index of refraction at the same time. So this is part of the objectives. So to summarize what we talked about today, I tried to give you a review of our activities on miniaturized optical devices for sensing, both using microstructures and nanostructures for biosensing and imaging. We talked about optimizing the design, the fabrication, and the imaging aspects to create these miniature fluorescence biosensors. And we worked and demonstrated in a lab in a chip for microfluidics aspects. We demonstrated both sensing using index of refraction as well as fluorescence sensing. And we discussed a little bit, but not too much, our progress towards implantable optical sensors. And I hope I gave you an impression of how to use our technologies and understanding of the biology and the physics to be able to use the devices for an optimized biodetection. I would like to thank the students who did the work. Muhammad El-Baheri, who is a proud graduate of Waterloo, so he's now a graduate student in my lab. Human, who's a postdoc, and they did most of the work with the nano devices. Thomas Sullivan and Elizabeth Monroe worked on the fluorescent sensors, and Hart and Dean have helped in measurements in live animals. Shofan and Kai are the two undergraduates who work in our lab, and I would like to acknowledge these collaborations and the funding agencies and the companies that helped us along the way. So thank you very much, and I'm happy to entertain questions.
All right. The room is open for questions.
Yes?
Have you looked at the temperature sensitivity of your nanostructure sensor?
Excuse me?
The temperature sensitivity. If the temperature changes, how does your...
Correct. So we have looked into it a little bit, but not too much. We have two ways to address it. One of which is to create a reference sensor that would be only seeing the water-based solvent, but not the analytes. And you could make it very easily in the microfluidics because you could manipulate in parallel two devices. We're looking into more sophisticated techniques that take balance detection schemes, but we haven't done it yet. So these are two elements. On the long run, we're also looking into materials that have high thermal conductivity. And if you have a high thermal conductivity, the influence of the temperature of the analyte would be less influencing your sensor. So these are several approaches to see how do you solve it. But the easier to solve is really to create two structures, one nearby the other, and create a balance detection scheme, which we could do with this element.
Also, have you looked at, like when you add microfluidics, does your Q factor change because of the...
So we looked at it. So far, we did not see a big difference between when we measure it in air than we measure it in fluidics. Now, take into account that we're not in the region where the absorption, which is about 1,000 inverse centimeter, makes a big difference. We're in a quality factor between 1,000 to maybe 2,000. I believe when you go to 10,000 and above is where you'll start to see it. And there's some analysis. One of the references I gave is by Ian White, who is now in Maryland. And he has analyzed that when you get to 10 to the 5 and above, there is where you start seeing the absorption and the fact you have water as a lossy media making a difference. And a simple solution to it is take the same sensor and move below the 1400 nanometer absorption peak of water. So any device below, you would see a steep reduction in the absorption, which is part of the motivation that we did to try to work at around 800 nanometer with very similar designs. And we could use oxide nitride technologies that are transparent and still fabrication compatible to allow us to do that.
One last question.
Yes?
Your optical filter in the photodetector, is this a thin-film dielectric filter?
Yes. So this is a specialized process where we stop mid-fabrication, send the wafer to chroma. They use their own emission filter technology where they don't want to tell anybody how they do it, but it's used on thermal evaporation of dielectric stacks. And we needed to take their own process and optimize it because depending on the substrate, there are shifts. So we optimize it and then we take it back, do a lift off, and then the sensor is ready. So this is the last step on the whole process. Yes?
About the laser, semiconductor captive laser, I would like to know what is the laser spot for this device, and you said that they are fully available. Can you give me a source?
So we are using, in this case, we made the lasers our own, but the epitaxial layer was done by Vixar, which is a startup company that was founded by Mary Hibbs-Brenner. So Mary used to be the head of the Optoelectronics Center in Honeywell, and then she moved with some of her people to open a startup. So they have the capability and the knowledge to create the epitaxial layer that gives you about 1 to 2 milliwatts in the region of 670 nanometers. You could buy the laser alone. You could not buy an epitaxial layer that has laser and detector. So we had to work on a custom design where we designed it together with her engineers and then went ahead and processed it all together. And part of my visit here, I hope to talk to Vito about what would it take to process these lasers here in Waterloo. You could make it single mode or multi-mode. So if it's a single mode, it's a diffraction limited spot. You could start with an oxide aperture that is between 4 to 10 micron. So 4 micron will be a single mode, 10 micron would be a multi-mode, and then you could focus it to a very similar spot. So it really depends on the oxide aperture in your own design. You decide if you want to have it a single mode or multi-mode device. Yes?
It's a very nice talk. And just to have a couple of simple questions. In any of your cases, do the sensor need to contact with the liquid or don't have to?
So
Do the sensors contact with the liquid in the channel or not?
in a case of fluorescence sensing, the laser detector module could sit underneath. So you have the fluidic channel and you touch from underneath or maybe a millimeter away. On the index of refraction sensing, the sensing layer, the photonic crystal sense, has to be touching the water because part of the design is to have the water on it.
In that case, how are you going to deal with the corrosion or use some special material to make the sensor? So if you work with any kind of liquid, that bio-liquid involve any kind of samples that will touch with the sensor, certainly they will affect the sensor.
Correct. So if you notice, there is an external readout to the sensing layer. So in the case of index of refraction sensor, you would have an external readout. The laser detector part is still detached, but the sensing layer would be inside the water. So you decouple the biorecognition module from the readout module or the transduction module. So the laser detector is underneath, whereas the layer that makes the frequency changes or the transmission changes is touching the water. And that layer is done with dielectrics. So silicon nitride, this silicon dioxide are not influenced by seeing water or seeing blood whatsoever. You could clean it up very nicely afterwards.
You don't have any kind of biomaterial attached to the surface?
This, well, the biomaterial sits, the biomaterial would sit on the top surface. This is where you would locate down there. So through a series of washes, you would wash and you would incorporate the biomaterial on that top surface or in the middle. There are some groups, for example, the Cunningham group in Urbana-Champaign that created a sponge layer by CVD that is sitting on the surface, and this is where the biomolecule sits. So depending on where you want it, you can create the sponge selectively either on the top surface or in the middle of your voids. It's part of your oblique CVD technique that allows you to make it. So it's something you could control.
And another question here is, how do you comment? Can we use this type of sensor for two-phase flow? For example, you have some agents in droplets encapsulated with, not encapsulated, generated by oil film. So inside is aqueous. Do you think this type of sensor can do the measurements?
We asked the question, we haven't answered it. Meaning we're in the middle of experiments to try it, because there are people who use droplets of what they call digital microfluidics, which is basically that idea of a droplet moving around. So we...
Not necessarily to use digital microfluidics, they use electrodes.
Correct, but this is an example of that.
Yeah.
We haven't yet... my thinking is that if the element, meaning the droplet, is larger than your 500 microns of detection at any given time, you're either fully covering it or not. So at the time where the droplet is fully covering it, you could measure it. When it's not covering it, then you're not measuring anything because there is an air bubble in the middle. So as long as you're covering the detection region, which is about 500 microns in diameter, you could make the measurement.
Okay, has to big enough to cover the entire detection region.
Yes.
I see the difference. Thank you.
All right.
Quick question. I really enjoyed your talk. Can I ask, you mentioned the many years of work in developing the device and the fabrication. Which aspects of fabrication were the most challenging to your group?
The CMOS compatibility. The ability, if you figure out a process and you want to work, and then comes somebody and says, step number 10, you can't get there because somebody else needed for a CMOS compatible device, therefore you'll contaminate the machine. So this has been the biggest challenge, to always go around it. And part of the solutions are to trying to find robust processes that we could still do without getting into that CMOS compatibility issue. So that has been, in my opinion, the biggest slowdown or roadblock in our designs.
All right, are there any more questions? We are already running pretty late, so let's thank the speaker once again.